It's a great pleasure to be here, and I'm not sure what I'm doing here, actually, because I don't really know very much about either virtue or fortune. But I do know about finding things out. And this is what makes me happy. So I hope this evening that I can give you some clue about how scientists find things out, and if possible, how this makes them happy. So this talk really has three parts. It has a preamble in which I want to explain how some things that are very well known are very difficult to explain and are not understood by me. Then I have a little bit of a biology lesson, background, I hope, for those of you who had your last biology lesson many years ago. And finally, a little explanation of what I discovered and why it made me so happy. And that's probably the most difficult thing of all. So let's start with this excellent mathematician who I once heard give a talk, Robert Aumann, who explained that he liked to work on problems that were absolutely useless. And I was very taken by that, and it turned out that some of his, what he thought were absolutely useless mathematical formulae became extremely important, and actually he was a pioneer, I think, in game theory, and which is important in economics. But here he speaks in his Nobel autobiography about science, and I really like what he says. Science is exploration, exploration for the sake of exploration and for nothing else. We must go where our curiosity leads us. We must go where we want to go. And eventually it's sure to lead us to the beautiful, the important, and the useful. Very interesting. Very often, in my experience, if one tries to study a difficult problem head on, you make no progress. And the way forward is to try and sneak up on it from behind. You can only do what you can do. And that's hard to explain to politicians. So now the real problem comes, and this comes from another, and in this case a Nobel Prize winning physicist who alas is no longer with us, but he wrote a lovely book called Soft Interfaces. Soft interfaces is about friction, how things stick together. And it's a beautiful little book, and it explains why things stick together when you try to move them. And right at the end, he talks about the differences between art and science. He says, science is clearly a form of art, same invention, the same doubts. There are major differences, however. One is the difficulty of communication. An Indian playing his flute in the streets of Bogotá invents a new tune, and within 10 seconds, passers-by may be struck by it, possibly for their whole life. But in our trade, a beautiful discovery can be transmitted only to people who have been through a long, specialized education. We must do our best to keep in contact with our fellow citizens, but we often fail. And I think that's a very nice way of exactly putting the problem of conveying the beauty. In other words, a scientist responds to another scientist's beautiful ideas exactly the same way you might respond to a concerto by Mozart. But in order to reach that point, you have to be in the same kind of space. And let's have a look at that, the difficulty of communication. Oopsie-daisy, where am I? Here we go. This is my daughter, Celia. I hope. She is holding Schrödinger's cat. The cat was called Minky. And 1 week after this photograph was taken, I'm sorry to say, Minky was eaten by a fox. So this poor kitten is no longer with us. Now I show you this because Celia asked me a very peculiar question. I had just had a phone call from Stockholm saying, "You have won the Nobel Prize." Imagine that. And about 2 weeks later, Celia asked this question, "Daddy, why is the ceiling opaque?" It's a strange question for a 7-year-old girl to ask, I think you'll agree. So I looked at her ceiling and I thought, well, it's opaque. Light does not get through the ceiling. But then I looked out of her bedroom window and a horrible thought struck me, which was, here am I, supposedly one of the greatest scientists in the world, and I have no idea how the light passes through the window. So I began to think of all kinds of theories about how light might pass through the window, through the glass, and I realized that some other things let light through. For example, air lets light through. The water lets light through. Alcohol lets light pass. So what differentiates these transparent materials from the opaque ones? And so I began to ask people, and sometimes they would say, well, that's because these other things absorb the light, and I didn't find that. So what is it about things that absorb and things that don't absorb? And one of my first revelations was to think about the element carbon. You're all familiar with the element carbon. Burning carbon is what warms our atmosphere. Burning carbon is what keeps us warm in the winter. Burning carbon is what makes our motor cars go along the road. But carbon is an interesting element. Not only does it burn, but it also makes the black color of black paint. But not only think of candle smoke and the inside of an ancient cathedral blackened with smoke, but it also is the sole constituent of diamond. Diamond is pure carbon. So diamond is completely transparent, and yet carbon black is completely opaque. So that tells you, or it told me, that the problems were with the electrons, the way that the carbon atoms are put together. And I began to ask more deeply. So I knew that this had to do with the interactions of photons with matter. And I had a look at Einstein's famous paper in 1905, where he talks about the photon. By the way, at the end of his life, Einstein said, "Everybody thinks they know what photons are, but I certainly don't." So even Einstein could be puzzled. Anyway, he writes in the introduction to this paper, which gets too technical for me later on, according to the assumption to be contemplated here, when a light ray is spreading from a point, the energy is not distributed continuously over ever-increasing spaces, but consists of a finite number of energy quanta that are localized in points in space, move without dividing, and can be absorbed or generated only as a whole. And this explains, you see, why you can see the distant stars, which are light years away, distant galaxies even, which are, it takes light billions of years to reach us. If light was simply a wave, by the time these ripples reached us, they would be absolutely negligible. But they haven't. These things that we see have traveled that vast distance in time and space completely unchanged. It's absolutely amazing. And I thought, gosh, what a romantic idea that is, but what are these photons and how do they interact with the electrons in matter? So I began to ask people, and the final answer came, and a sort of final difficulty dawned on me when I sat next to a friend of mine at the British ambassador's lunch for the British laureates in Stockholm in 2001. And I said, Aaron, you know about these things. How does light get through glass? And he said, well, Tim, you really need to understand Schrödinger's equation. And when he said that, I knew that I was doomed, lost, because what is Schrödinger's equation? Here is Schrödinger. I like Schrödinger. He wrote a lot. He was very philosophical, almost religious, and a fine physicist. And here is his horrible equation. How does this explain how light gets through glass? It looks like a simple equation, but actually, when I tell you that I, with which it starts, is the square root of -1, I had great difficulty with that concept when I was a student. H bar, the next symbol, is Planck's constant divided by 2 pi. Psi dot is something called the rate of change of the wave function with respect to time. Now, I don't know what a wave function is. And that equals h, the Hamiltonian, that is Hamilton, and times psi, which is the wave function. Now, you will see, at least those of you close to the screens can see that there's a little dot over the right-hand psi. Terrific stuff. Anyway, but as far as I'm concerned, this doesn't explain anything, but it is the equation that explains it. Interestingly, the great Paul Dirac said the problem of getting the interpretation of this equation proved to be rather more difficult than just getting the equation itself. So even very, very fine mathematical physicists took them some time to interpret the true meaning of this equation, although the equation was absolutely correct. And I like this quotation from Richard Feynman very much. He together with Tominaga and Schwinger, won the Nobel Prize for working out the theory of QED, quantum electrodynamics. And he says, such an accurate theory of the interactions of photons and electrons that if you were to measure the distance between New York and Los Angeles, it would be specified to the width of a human hair. That means that this is one incredibly accurate, incredibly well understood to quantum physicists things. But, and it is a big but, apparently, I've asked physicists about that, they still don't really understand what happens when that wave function in the equation collapses and actually interacts with the electron. So in one sense, we understand how light passes through some materials and not through others. At least some people in the world do, but I don't. And it bothers me that such a simple question can have such a complicated explanation. And when it comes to things like electromagnetic radiation, I am completely lost. The great Clerk Maxwell interpreted Michael Faraday's results, and let's not go there. This is one of the great triumphs, I think, of 19th century science. But it's very difficult, and you have to be a superb mathematician in order to understand how it all works. But fortunately, there are people who understand these things, and actually, just this is really a parenthesis. Some of the greatest advances in medical visualization have come not from doctors, but from physicists. Röntgen, the guy on the left, was, of course, the discoverer of X-rays. Zernike invented the electron microscope. The phase contrast microscope, Ruska, the electron microscope, and Lauterbur and Mansfield invented the more modern techniques with which you can see inside bodies completely non-invasively and completely non-toxically. X-rays interact with matter and can do damage, whereas magnetic resonance imaging does not. So this is my mother-in-law's head. She has they thought she was, they thought she was depressed. They gave her antidepressants. And it wasn't until she began to fall over that they decided maybe she had a stroke or something like that, took her in, put in one of the machines and saw this amazing thing. It was the size of a grapefruit. And using the same visualization techniques, the surgeon cut it out. And here she is, 8 years later, perfectly well. Just cut it out. Amazing, absolutely amazing. In fact, she died a couple of years ago, but not from that tumor. She had another stroke. So back to Schrödinger, he asked in his question, this question in a little book that he wrote. It was actually a series of lectures that he gave, exactly the same year that I was born. In fact, not even the same year, but the same month that I was born, 73 years ago. How can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? And this was really a sort of rhetorical question, but he had faith, tremendous faith, that actually there was no such thing as a vital force, which many people would have believed at the time. He thought that everything obeyed the laws of chemistry and physics, and it was simply a matter of finding out how those laws applied to living organisms. So what can we say about this? Well, actually, during my lifetime, this has been worked out in tremendous detail and is just like the theory of quantum electrodynamics. I think we can say there are no mysteries. There are many puzzles. How the brain works, how we see things, how we hear things, is still deeply, deeply mysterious. But basically how life works is not. And I want now just to briefly, as briefly as I can at least, give you an impression of what I think life is like. So the atomic theory of life is that everything is made of cells. Very, very small. You have, each one of you, about 5 times 10 to the 13th cells. That's, I don't know how you call that, 10 trillion cells each. And this has been known for a very long time, even before it was known what a cell was or the significance of it. Botanists saw, in this case, it's a plant, the stem of a plant, and you can see that Nehemiah Grew, who was a distinguished, he was actually a doctor in the late 18th century, 17th century, clearly understood that plants were made of cells, but he didn't know their significance. He didn't know how they came to be, or how they were generated, or how they went away, how they changed their shape, or anything. And it wasn't for another 200 years almost that it became clear that animals are also made of cells, just like plants. And it also only very gradually dawned on people that cells only ever come from other cells. As far as I know, there has been, since the original creation of the original cell, and we don't know where that cell came from. Maybe it came from outer space, maybe it was a lucky accident in the primeval soup on Earth, I don't know. But from that original cell evolved all other cells and all other organisms. Obviously, the invention of cells sticking together to make larger, more complicated organisms was a great moment in evolution. And most of the kingdoms of life are these things like on the left, the bacteria, which are the kind of bacteria that cause disease, the kind of bacteria you find inside you, the kind of bacteria that cause food poisoning. In the middle, archaea, which look like bacteria, but they live in much more hostile environments, generally. Things like hot places, salty places, acid places. And on the right, you can see us, the multicellular organisms with a proper nucleus. The word karyon means nucleus, a kernel. And you can see this line of descent that goes back to the original cell. But this raises a very interesting paradox, because as I'm sorry to say, none of us are going to live forever. So it's curious, isn't it? We can trace back our ancestry maybe 2, maybe 3 billion years, and yet we are all mortal. So what is it that actually is continuous? And the answer is, and this became clear at the end of the 19th century, the answer is that it's our eggs and our sperm that are immortal. They carry on into the next generation. We have children, and somehow every generation there is a renewal through a testing of the eggs and the sperm to make sure that they make a good baby. And it's as though we ourselves are merely fruiting bodies. I mean, it's a very sort of funny idea, to think of yourself as a mushroom or a flower. So let me show you how cells divide, and these are bacteria. Whoops. Why is this not going to work? That's interesting, at least on my screen. A moment. So there you see cells, bacteria multiplying by division, and one can write that down as this sort of little diagram. One cell splits into 2 cells, the 2 daughter cells then grow up, and they repeat the process. So 2 cells becomes 4 cells, 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, and you can see that exponential increase very scarily. Now, what do our cells look like when they grow? This is what they look like. Tensing. Something goes on inside and then they relax. Now, what is that something going on inside? That again was seen a long time ago. 1882 is the publication. And you can see here the things in the middle called chromosomes. And I'm sure everybody knows what a chromosome is or what it looks like. They're called chromosomes because they stain with colored dyes. That just means a colored body. And you can see they're lining up in the middle of a cell, and you can make out roughly that there's something, which we'll come back to, which is holding them there and which is going to pull them apart. And the process of division can be divided into various stages which have names that school children have to learn, but it's really not relevant to the process. The important thing is that let's go straight to it and not worry about getting into mitosis and getting out of mitosis, but go to DNA, because DNA is the key here. And DNA, as you know, has 2 strands. Interestingly, those strands run in opposite directions. One runs up and the other one runs down. And it was early on that the central dogma was enunciated. Francis Crick said that he thought a dogma was something that you just had to believe, but there wasn't very much evidence for it. And it was absolutely right. DNA makes RNA, makes protein. and proteins make cells, and cells make organisms. Let's see what this really means. This is one of my favorite exhibits on the Euston Road in London in the Wellcome Museum of Science, and it is simply a printout of the human genome, which is now almost completely worked out. Now, as you can see, this printout is divided into volumes because the chromosomes have numbers. Here you can see chromosome number 10, 11, 12, 13, and below 15, 16, 20, 21, 22, and 23 of them altogether. What does one of those, the page is open on the page. This is what it looks like. There are the instructions for making a human being. Actually, this is only the instructions for making one of the chains of hemoglobin, the protein that carries oxygen in your blood. And you will see that there are no punctuation marks, there are no paragraphs, there are no chapters. Somehow the cell knows exactly how to make use of this information. And the amount of information is absolutely staggering. The complete works of Shakespeare, I found out from the internet, Google, has roughly 3.5 million characters. But the human genome has 3 billion characters. So each one of your cells, of which you have an astronomical number, has 812 complete works of Shakespeare's worth of information in it. Isn't that amazing? Only the red cells have thrown away this information because they're going to die young and they don't need it anymore. They never replicate. And the key thing is that every cell must contain this information because it's like a library from which you can ask the question of what must I do in certain circumstances. So the key to cell division is actually first replication of the DNA, that is to say the two strands of DNA separate and are copied, hopefully flawlessly, and then you can see the chromosomes come into being, and the chromosomes, one chromosome goes to one cell, the other chromosome goes to the other cell, which means that the two daughter cells of division have exactly the same information in them with no mistakes. And here's a little movie that I found on the internet, and I paid $15 for it. It contains one mistake, which I won't go into, but hopefully gives you the idea. Oops. Is that going? Yes. So you can see the chromosomes lining up and then getting pulled apart and going into the two daughter cells. Think about that. And this is my ex-boss, well, actually, I suppose he's my present boss too, Paul Nurse's diagram. So the key bits are replication of the DNA in one phase of the cell cycle and segregation of the chromosomes at the other. And this is what the cell cycle is all about. Basically, two phases, replication, segregation, separated by gaps, which are called G1, gap 1, and G2, gap 2. And in this cycle, it's the thing that particularly interests me. There are 3 switch points when, if everything is okay, the wheel can continue to turn. But if something is wrong, the wheel stops turning because it is so important that the two daughter cells inherit a complete set of accurately copied chromosomes. So this obviously raises a question. I mean, this is an absolutely fundamental and important biological process. So how does it work? And in particular, how is it controlled? So I did not set out my career to ask that question, but it was a question obviously lurking always in the background, and every so often somebody would make a small advance in understanding, which I followed. And then the opportunity came to go and work in this place, Woods Hole, Massachusetts, which is about 2 hours drive from Boston, as you can see, rather like here. It's a seaside place. and it contains 2 laboratories and not much else. It has a bank, a couple of restaurants, a bar, actually 2 bars, and I went there by sheer chance. I lent somebody my bicycle and we became friends because I lent him my bicycle. And the idea was to work on sea urchin eggs and to study protein synthesis. I was an expert in the control of protein synthesis. And the important thing about that is that when sea urchin eggs are fertilized, there is a big increase in the rate of protein synthesis. I wanted to understand that switch. So here's a female sea urchin in black and white. Here is a male sea urchin in color. They both taste equally delicious. As you can see, the sperm is white and those eggs are a dark red color. Now, what happens if you mix the eggs with the sperm? So this is pretty exciting because you can do this in a test tube. You can just do it in a beaker if you're present in Woods Hole during late June and most of July. Let's see what that looks like. Hopefully that will start the movie going. As you can see, if you mix the eggs with the sperm, I won't say that you get a baby sea urchin because that wouldn't really be true. It takes about 2 years to get a fully formed baby sea urchin. But you can see that one big cell has become now a hollow ball of much smaller cells. Now, a key fact which I knew and had been known for a long time is that if you stop the protein synthesis that interested me, sea urchin eggs stop dividing. So remember that. I mean, if you stop our cells dividing, stop protein synthesis in our cells, they can't grow. So of course they don't divide. But it's a little bit different with sea urchin eggs because they're very large cells and they already contain almost everything they need to divide. But clearly they need to make something new in order to enter division. So that was sort of in the, it's one of those things that nobody really thought very clearly, deeply about it. And it actually, in biology, very often such thoughts there are so many different possible interpretations that it's not terribly useful. But it is an important fact. And then one day, this guy here, John Gerhardt, came to give a seminar in the course that I was teaching. And he explained, and I'm going to show you hopefully this movie. Look at the right-hand things are not sea urchin eggs, but frog eggs. And he explained how progesterone catalyzed a cell cycle transition in the frog eggs. And if you look very carefully, not a very exciting film, but I hope. So look underneath that white spot, and what you find is that actually you start out with a cell with a big nucleus, and you end up with a cell with chromosomes lined up. So the progesterone hormone has caused an entry into the cell division process. And what John described was experiments which had been done a few years before by a Japanese postdoc working in Yale, which showed that actually what the progesterone did was to activate a mysterious inactive enzyme that was already present in the oocyte and just needed something to turn it on. And you could plot the comings and goings of this substance, and it followed exactly the cell division cycle. That is to say, you could find this magic factor. The magic factor is called MPF, for maturation promoting factor, and it's always high in dividing cells and always low in cells where the DNA is being replicated. And actually, it's not just in eggs, but it's also in the rapidly dividing cells. And in fact, eventually it was discovered that all cells, our cells, contain MPF, only when they're in the act of division. You remember that early movie where you saw the cells all round up, tense up? They have MPF, and when they relax again, the MPF has gone. So the problem with this was that we didn't know what MPF was, and if you tried to purify it, it disappeared. It was something that was terribly, terribly unstable. So you didn't know what it was, you couldn't tell what turned it on, and what made it go away was equally mysterious. So this is clearly a centrally important substance, but full of mysteries in every direction, but seemingly the key to cell division. It looked like the enzyme that catalyzed cell division. I thought that was a crazy idea. I mean, I was captivated by it, because enzymes are things that digest your dinner. Enzymes are things that allow you to breathe properly. Enzymes can make you move your arm. But the idea that an enzyme could catalyze cell division struck me as quite extraordinary. And what kind of an enzyme could it be? Well, you know, this was just an idea. 1979, that seminar was. And it wasn't until 1982 that I did another funny experiment. Now, let me tell you about this experiment. So the impulse for the experiment was an interest from reading this book, which you see is by a very distinguished late 19th century biologist called Jacques Loeb, called Artificial Parthenogenesis and Fertilization. I was reading this book because he had worked a lot on sea urchin eggs, and he described experiments where basically you could get the eggs to develop without sperm. Things like dilute acids, dilute alkalis, dilute solutions of soap. All these things could set the development of the egg in motion. And I began to wonder about patterns of protein synthesis. Were the parthenogenetically activated eggs the same or different from properly fertilized eggs? And to some extent, this was also sort of, you know, I sort of liked this idea because I had had a very religious upbringing. And I can remember a time when to question the birth, the virgin birth of Christ, would have been to me the most terrible, undermining of my faith. I mean, it was an article of faith that Christ was born by virgin birth, as we see in this beautiful Fra Angelico picture. You can see the Holy Ghost impregnating the Virgin Mary, and the angel announcing the fact. So I was sort of, this was, by this time I had lost my faith, but I liked the idea that one could do a molecular experiment on religion, I think. Anyway, so I did the experiment, and I compared these patterns of protein synthesis, and what happened was that I stumbled on the most important discovery of my life. And this is it. This is a Nobel Prize winning picture. Now, most of you probably haven't looked at very many of these pictures, and I think this is where the problems in communication come in quite difficult. So let me just go and I tell you, this is a one-dimensional SCS polyacrylamide gel. This is not going to help, but it is. You see these black bands here? Each one of those black bands is a radioactive protein with which I use to trace the thing, okay? And I want you to focus on this one. You see it comes and then it goes, and it comes and it goes. Okay. So that one protein labeled cyclin, it comes and then it goes. It comes and then it goes. So I saw this protein disappear. That was the discovery. And I suddenly realized that the comings and goings of this protein precisely mirrored the comings and goings of MPF. There were a couple of problems, and you can see it much more clearly here. In fact, there are two of these proteins, it turned out, when we looked at another organism. This is from a clam, not a sea urchin. And it was true in clams too, that if you prevent the synthesis of these proteins, the cells cannot divide. So the very simple hypothesis arose in my mind that you had to make something to catalyze cell division, and then amazingly you destroyed it to complete the cell division. And that was it. That was the Nobel Prize winning idea. Now, I mean, there are all kinds of problems about this. I mean, if you look at that, you can see that actually cell division is very abrupt, and the rise in this protein is very gradual. And I think I can put an analogy to explain that in your mind. You know, a light switch. You imagine a light switch which is on or off. When your hand moves that switch, the switch moves slowly, but the switch actually switches quite abruptly at a certain point because of the mechanical device is inside. And it turned out, after a lot more work and many, many more years, that actually this is absolutely correct, and that the enzyme that catalyzed cell division was a thing made of two parts. One part on the left is called CDC2, or sometimes CDK1, which is an enzyme which absolutely requires my protein, the cyclin, to turn it on. And basically what the enzyme does, here we see the process, in order for a cell to round up, you have to stick all these phosphate groups onto hundreds of proteins inside the cell, and that addition of phosphate groups is done by the enzyme that you see above the arrow in the middle. And there are, as I say, hundreds of these substrates. So what had previously been completely mysterious and impossible to purify, don't know what turns it on, don't know what turns it off, suddenly became absolutely clear, very simple when you understand. The cyclin accumulates, CDC2 turns on, and the proteins get phosphorylated, and the cell enters mitosis. And you see this dramatic change in the cell that you saw in that movie. They round up, they release themselves from the substrate, the chromosomes condense, and the nucleus disappears, and so on and so on. Lots of things change, and that's brought about by this phosphorylation. Then the cyclin is degraded. The enzyme turns off because it absolutely needs the cyclin to stay on, and the cell relaxes again, and cell division is completed. So it's a perfect example of, it's a very good example actually of a sort of Nobel Prize winning discovery, because previously people would have completely disbelieved the idea that a protein could disappear like that. Because enzymes just can't be that specific. They can digest all of the meat that you eat, but you couldn't digest just one protein in a cell without digesting all the others. But there I saw it. You've seen it for yourselves. It really does. It's very, very, very specific. So now we just briefly consider, we now understand cell division. Do we therefore understand how cell division goes wrong in cancers? And here is a sailor with a very nasty tumor. I found him in a medical museum in London. And you can see that what's happened is that the cells of his lip are just growing totally out of control. And people said, well, now you understand how cell division works, you should be able to stop the cell division in tumors. But the problem is that the tumor cells are very good at dividing, and if we stop them dividing, we also stop our normal cells dividing. And if you stop your normal cells dividing, you die very quickly, in about a week, in fact. Your stomach lining cannot replace itself, your blood cells cannot replace themselves, your hair falls out. It's extremely unpleasant. And basically what happens in tumors is not that there's something wrong with cell division, it's something wrong with the organization, it's something wrong with that 812 volumes of the works of Shakespeare's worth of information. If you look at an actual, here's a micrograph of a drawing actually of a kidney tubule, and you can see this is made of cells, but it's tremendously complicated. And the cells at some point can form branches, the cells can form narrower tubes. Up at the top is a blood capillary, which is also made of cells, but very different cells from the ones that make up the tubule. And the problem in cancer is that the information is lost and the cells can no longer organize themselves. So instead of these cells on this poor guy's lip looking like a lip, they now just look hopelessly disorganized because they've forgotten who they are. And it's in finding out exactly what is keeping those cells alive, what they have forgotten, that maybe future advances in understanding the treatment of cancer will come. But for the time being, the remedy is best, as was done with my wife's mother, to cut out the tumor while it's very, very small, because it's very difficult in most cases to find particular things in tumors that aren't also present in normal cells. And chemotherapy, as you well know, although it's effective, it's still a pretty unpleasant and rather a blunt weapon in the armory of cancer doctors. So hopefully things will improve along those lines through better understanding of how cells come to be exactly what they are, how cells form tubules and things like that. So the important message here is that finding that protein was the most thrilling thing in my entire life. I was so happy when I discovered that, because I knew that I was the first person to understand the basis of cell division. And telling other people about it was absolutely wonderful. Moreover, I knew that I'd stumbled into a fresh field that was much more important and much more interesting than the field that I'd come from. And I think it's important to emphasize that the experiment which revealed things was completely lucky. I did it, as I explained, for entirely the wrong reasons. But I knew enough, because I'd been thinking about the problem for long enough, so that when that little tiny clue came, I was able to correctly interpret it. And that discovery then has kept me going for the rest of my life, and still continues to engage me. And if I had another hour, I could tell you about what me and my friends are thinking about now, but I think I have said enough. Thank you very much for listening.